Modern medicine has made incredible strides. We can now map the human genome. We can make artificial hearts that we put in the body. We can even put little robots through the body to clean up and repair pathology. But there's one thing we don't have. Apart from one or two exceptions, we can't really monitor the body's chemistry in real time on the timescale that it changes. For decades now, we have had continuous glucose monitors, and they have revolutionized the lives of people with diabetes. But even though we've had them for more than 20 years, we actually don't have similar technologies for other molecules. For other molecules, we're still in the dark. And so what we do is we take blood tests, which take an hour or more to perform. And so our medical decisions are based on knowledge of where you were, not where you are now.
What's the best way to measure molecules in the body? Well, I'm a biologist by training and a bioengineer by profession. So not surprisingly, when faced with that question, my research group and I turned to biology for inspiration. We looked at the naturally occurring chemoperception systems, the mechanisms that organisms and cells use to detect specific molecules. In looking at those, we found that there's a commonality amongst all of them. In every case, evolution engineered the receptors that recognize the target such that they undergo a large shape change upon binding that target. That shape change is then used to transduce the recognition event into some output signal that the cell or the organ or the organism can use. Now, having an inspiration is the first step. Coming up with a way of translating that innovation, that inspiration into an innovation, into a real technology. To achieve that, the first thing that we had to do was come up with a way of re-engineering receptors, of artificial receptors that we could use in our technology, such that, like the naturally occurring receptors, they'd undergo some big shape change upon recognizing their target. Specifically, what we did was to show that in a class of artificial DNA-based receptors called aptamers, that we could re-engineer those so that they only folded up upon binding their target. That is, they shifted from a flexible, floppy, expanded polymer into a well-defined three-dimensional structure upon binding the target that they recognize. We then developed an electrochemical method of monitoring that folding event, which then in turn reports on the presence of the target. Putting all of this together, we came up with our technology, electrochemical aptamer-based or EAB sensors. These are continuous, and because they use the same mechanism biology employs us to perform chemoperception, to perform molecular recognition in us, our sensors work in situ in the body.
This was an immense discovery in biosensing. It completely transformed what we can do. We now have the potential on a single platform to monitor many different molecules in the body in real time. We've never been able to do that before. It takes the incredible power of the blood glucose monitors into a whole realm of other molecules of medical interest.
One critical and immediate application of this technology is to inform drug dosing. Take, for example, the life-saving antibiotic vancomycin, which is notoriously difficult to dose appropriately. Too little risks treatment failure, and too much can potentially harm the patient. Dosing of critical care medicines like vancomycin is currently based on population averages and not the individual. This can be particularly problematic for critical care settings such as the ICU. Lab diagnostics are not enough because they only provide a snapshot in time. By the time we get the result, the patient's physiology has already changed. Often, biology moves faster than we can. By offering clinicians real-time continuous insights into drug levels, EAB sensors solve this problem, replacing limited and delayed data with continuous real-time data, dynamic dosing, and truly personalising care.
Bayesian software proved that personalised dosing software can significantly impact patient care, reduce adverse events, and improve outcomes. And Bayesian dosing worked by taking tools that we already have in hospitals and just using them in newer and more innovative ways, doing better with what we have. What makes EAB sensors special is that it's a new technology, but one that fits directly into current clinical care and current practice. Bayesian dosing provided an estimate. An EAB sensor provides a real-time, continuous monitor of the marker that we wish to measure. It brings the pathology lab to the bedside, allowing us to make a more accurate and more rapid diagnostic decision and improve patient management. Vancomycin is only the beginning. Whether it's measuring hormones, biomarkers, or other drugs, an EAB sensor platform allows us to rapidly do real-time monitoring of so many markers that are directly relevant to patient care.
Having extensively validated this technology on the lab bench and in animals, commencing human trials. These human trials are proving the safety and efficacy of this fundamentally new way of monitoring health.
Rather than relying on laboratory results delivered hours or even days later, now clinicians and their patients can base dosing on real-time continuous information.
With technology like this, we could have a patch on every critically ill patient in the hospital. Soon we'll wonder how we practice medicine without these continuous insights.